Welcome students to lesson 2 of cell, which is cell division. We are looking at meiosis. And meiosis involves 2 divisions of the parent cell, resulting in 4 daughter cells. The mother cell has the diploid number of chromosomes. The 4 cells or the gametes have half the number of chromosomes. They are haploid. So when you say the mother cell has diploid number of chromosomes, that is, they are 2n. But the daughter cells or the 4 cells have the number of chromosomes. They are said to be haploid. So 2n divided by 2 or half, you will get n. Meiosis consists of 2 successive divisions. In the first meiotic division, the parent cell splits into 2 and the products divide again to form the second meiotic division. So after the parent cell has divided into 2, that forms the first meiotic division. So these 2 cells from the parent then divide to give second meiotic division. And in the first meiotic division, the homologous or sister chromosomes get separated from each other and move to different cells. But in the second meiotic division, chromatids separate. So when it's about the first meiotic division, it's about homologous chromosomes. But in the second meiotic division, it's about separation of chromatids. Meiosis is divided for convenience into series of stages, just like in mitosis, and each is followed by either 1 or 2, indicating whether it belongs to the first or second meiotic division. Let us look at what happens in first meiotic division. Interphase. So this is interphase 1. As in mitosis, the cell prepares for division, and there is multiplication of chromosomes, synthesis of new organelles, and build-up of energy to drive the entire meiotic process. So this is there are no visible chromosomes in the nucleus, and the organelles are being synthesized. So just remember that this meiosis takes place in reproductive organs, whereas mitosis occur in body cells, also known as somatic cells. So the second stage is prophase 1, and here the nucleolus disappear. Then centrioles, if they are present, become arranged at the opposite sides of the nucleus. So if the nucleus is here, then centrioles would be at the opposite side. But in this case, we do not have the centrioles. Homologous chromosomes lie side by side through a process called synapsis to form bivalents. So these are homologous chromosomes. They are lying side by side. So these are the bivalents. The chromosomes become short and thick, and then they become more visible. But in interphase 1, there were no visible chromosomes in the nucleus. Chromosomes may become coiled around each other, and the chromatids may remain in contact at points called chiasmata if there are many. But if it is at one point, then it is called chiasma. Chromatids cross over at these points called chiasmata, and that allows exchange of genetic material. We have the third stage or phase called metaphase 1, and during stage the nuclear membrane has disappeared. The spindle fibers are fully formed and attached to the centers or the centromeres. The bivalents, these ones, move to the equator of the spindle fibers, the equator or the center. Then during anaphase 1, we have homologous chromosomes being separated and then migrate to the opposite poles, and then the spindle fibers become short. So the homologous or the sister chromosomes are at the poles here. We go to telophase 1, the last phase. The daughter cells go into a short resting stage called interphase. Then sometimes chromosomes may become condensed and the daughter cells go straight into the phase of second meiotic division. So they are resting. This is what had already occurred during anaphase 1. So then here there is only rest of the cells. We move to the second meiotic division, and the purpose is to separate chromatids from one another. And remember, we said first meiotic division separates chromosomes or homologous chromosomes, and then they are moved to different cells. So we move to interphase 2, and here the daughter cells go into a short resting stage that is called endophase, jumping from telophase 1 to interphase, that is rest. Then during prophase 2, new spindle fibers are formed. Remember, previously there was rest, so we have formation of spindle fibers. Then in metaphase 2, the chromosomes move to the equator of the spindle fiber. After that, the chromosomes orientate or arrange or position themselves towards the opposite poles. So you see the chromosomes are at the equator, but at the same time they are positioning themselves at the opposite poles. Then during anaphase 2, sister chromatids separate from each other and move towards the poles, pulled by shortening spindle fibers. And finally, we go to telophase. Then spindle fibers disappear and the nucleolus reappears, and as well as a nuclear membrane is formed around each set of chromosomes. Then what follows is division of the cytoplasm across the middle and the 4 daughter cells. So already we have 2 daughter cells from meiosis 1. Then during the last stage of telophase 2, these 2 daughter cells from meiosis 1 all the way will divide each by 2, so that we can have 4 daughter cells, which are also called tetrad. And this cell, each one of them is haploid. So we are saying each one is having half the number of chromosomes from the mother cell. So we have 2, 1, 2, 3, 4 tetrads. So we are done with the first and the second meiotic divisions. We want to look at importance of this process of meiosis. So we say meiosis brings about formation of gametes that contain half the number of chromosomes as the parent cell. We have all said, that the mother cell, if the mother cell is having a diploid number of chromosomes, then during division or meiosis, it results into haploid number of chromosomes after division. It helps to restore the diploid chromosomal constitution in a species during fertilization. So for instance, if during fertilization we have this gamete which is having haploid, and then this one is also having haploid. So when you combine an N from here and an N from this other side, we get 2N, which is the diploid state. For example, we have said human beings is having 46 number of chromosomes. So during meiosis, and we have said meiosis takes place in reproductive cells. So the male will produce 23 chromosomes and the female will contribute 23 chromosomes. When they fuse, then the diploid state, that will result in 46 chromosomes in the zygote. So then what are the similarities between mitosis taking place in body cells and meiosis taking place in reproductive cells? Remember, both take place in plants and animal species. Equally, plants have reproductive organs, which is the flower. Then both stages, or both involve division or multiplication. As we have said, we have anaphase, prophase, and all those stages, and in each there is division or multiplication. Then it brings about new gene combination that lead to genetic variation in offspring. Just as I had mentioned, assuming this is a cell from the male and this one from the female. So when these two combine, they are different, then that combination leads to genetic variation. And this combination, if there is the variation, means there are differences among the offsprings or the young ones. And these variation or differences, sometimes they are good, sometimes they are bad. In genetics, we shall discuss about these genetic differences in detail. Then we want to compare now the differences between mitosis and meiosis. So in meiosis, we have the homologous chromosomes associating with one another. But in meiosis, homologous chromosomes do not associate with one another. Then meiosis occur in two nuclear stages, each in two nuclear division, each having two stages. So we have meiosis 1 and meiosis 2. But then mitosis occur in one nuclear division of 5 stages, and this one is having 5 stages. Then meiosis produces 4 haploid daughter cells, but mitosis produces 2 diploid daughter cells. And we have all said then meiosis occur in reproductive organs, but mitosis occur in body or somatic cells. Also, there is chiasma formation, which occur leading to crossing over that leads to exchange of genetic material. But in mitosis, there is no chiasma formation, therefore no crossing over. And then if there is no crossing over, then no exchange of genetic material. We come to the end of the second lesson. Quiz yourself. Then the answers are within our notes. You may replay the video so that you can get the assignment to capture all the questions and even the answers. Goodbye.